now? Okay, so we will start. Now see, four chapters were given to you for the preparation. One chapter was the cell, second chapter was the biomolecules, third chapter the cell cycle, and your fourth chapter was the cellular respiration. Okay, now I will be asking maximum 5 questions to each student, and each question will be carrying 2 marks. Okay, and from each chapter, you find that one question will be asked, and from your cellular respiration, I will be asking 2 questions to you. Is it clear? So total that way 5 questions will be asked, and if you are able to answer all those questions which I have asked, then further I will not ask any question. Okay, अगर ऐसा होता है that you are not able to answer any question at the end, one by one I will ask from each chapter. And at the end, then if I find that you are not answering any question, so I will be giving you 2 more questions. Okay, and if you are answering them, so then you can understand that you will be getting the 50% marks. ओके और नहीं तो फिर आप समझ जाएंगे कि कैसे करेक्टनेस के ऊपर मार्क्स रहेंगे, योर एक्सप्लेनेशन, your clarity of expression, your concept, how clear they are. Okay, then your communication skill and how attentively you listen my question and understand, all that will give you the marks. So this way the 5 parameters are there, children. One is clarity of concept, another one is the clarity of expression, then your communication skill, attentiveness, and comprehension. Okay, so you understand my question which I asked and you are explaining me well, so then you will be getting the marks accordingly. Followed all of you all? So I hope this is very clear to each and every student. So we shall start the oral test for today. So first Neha is ready. Now the question for you from your cell chapter is, tell me what are the important components, structural components of the mitochondria. The structural components of mitochondria.
The structural components of mitochondria is that mitochondria has a two-unit membrane, the outer membrane and the inner membrane. The inner membrane is projected in a finger-like projection, forming the cristae. On the surface of the cristae, we will have F1 particles are present, which are also known as oxysomes.
Very
And in
good.
the inner membrane encloses a matrix which have its own DNA, ribosomes, and other molecules.
Okay. And can you tell me the function of each part, each component?
The F1 particles, they help in the electron transport chain as they act as there is a site of ATP synthesis, and in the F1 particle there are two regions, the F1 region and the F0. F1 acts as the ATP synthesis site and F0 acts as a tunnel for the transport of protein. And then as mitochondria has its own DNA and ribosomes, it is known as semi-autonomous organelle also. And the membrane protects it from the external environment. It keeps it within the place in the cytoplasm of the cell. And mitochondria is the powerhouse of the cell. It helps in the oxidation of food and production of energy, which is stored in the form of ATP.
Very good. Okay, correct answer. Now the next question from your biomolecules. Now tell me which bonds are formed between the amino acids to form the protein and how?
In amino acids, peptide bonds are formed. In that, like one amino. Yeah, the amino acids are bound by the peptide bonds, which are like the amino acids may be linked one after the other, forming a long chain, and these chains are held by the disulfide bridges, or they are attached in a
Huh. First, tell me that how the peptide bond is formed. Peptide bond formation.
peptide bond is formed by the process of dehydrogenation in which a H2O molecule is released in that,
Okay.
and
So between which
like the
function
between which
means amino acids, how they are joining.
it is formed between the carboxyl group and the of one amino acids and
Correct.
the amino group of the another amino acid.
Correct. Okay. And then it will form a protein. So what are
Protein.
the conformations of protein? Name them.
There are four conformations:
Huh?
primary, secondary, tertiary, and quaternary.
Okay.
In the
So, hmm,
Ma'am, in the primary conformation tells the sequence of amino acids in which they are linked by the by the peptide bonds. The
okay.
secondary structure shows two types of model: alpha helix and beta pleated structure. In the alpha helix, there is only a single polypeptide chain which is folded upon itself, uh, and then the hydrogen bonds are present, which keeps it stabilized. And in the beta pleated structure, a plate-like structure is formed in which more than one peptide chains are present, which may be folded upon itself or may not be. In
Uh,
the tertiary structure, we have in the tertiary structure the they are attached. And I'm first telling the quaternary one. In the
no
quaternary
problem.
structure, the polypeptide chains are not linked. They are they are present independently. But when they come together, they become active, or else they stay inactive.
Uh,
And the tertiary structure, they
so it gives the 3D structure now.
3D
Okay.
structure. Yes,
3D structure we'll get it in the tertiary and quaternary.
ma'am.
That will be
Yes,
formed. Okay. So correct. Now cell cycle. Tell me the significance of meiosis. Significance of
ma'am.
meiosis.
Meiosis.
Significance of meiosis. Tell me that.
Ma'am, in meiosis is important for genetic variations because crossing over takes place, and in meiosis, four daughter cells, like from a parent cell, four daughter cells are formed. It mostly takes in the reproductive cells or germ cells. In this division of like reduction of chromosome numbers takes place in order to maintain the chromosome number in the progenies, and uh, they like it does not uh, due to crossing over. There are like when the four daughter cells are formed, two will be of parental type and the other two will be the recombinants.
Recombinants are there. Okay. Just tell me where this crossing over occurs, in which stage?
It occurs on the chiasmata, it in the prophase one of the paquitin.
Pectine
In
stage,
prophase
prophase one. Correct. Very good. Okay. Now the next question. From the respiration, now I'm asking. Tell me that what are the steps involved in the cellular respiration?
in cellular respiration, steps involved are: first is the glycolysis, in which six carbon compound is broken down into a three carbon compound, which is pyruvic acid. Krebs cycle: after pyruvic acid then undergoes oxidative decarboxylation. Then the acetyl coenzyme A is formed, which is a two carbon compound, which enters into the Krebs cycle, which is also known as citric acid cycle or tricarboxylic cycle. And then in the last process is the electron transport chain.
Electron transport chain. Okay. So when one glucose molecule is oxidized through all these steps, so how many ATP are produced?
If one glucose molecule is oxidized, 38 ATP or 36 ATP are produced.
Are produced. Okay. Now, can you name the electron carriers involved in this ETC?
NADP, NADH, or FADH, and the cytochromes B, C, A, and A3.
A3. Okay. So, what is the role of these electron carriers?
These electron carriers carries the electrons through the FAD and the cytochromes, and like the energy that is produced is used to transport the protons through the membrane. Thus, a proton gradient is created across the membrane. We know that the membrane is impermeable to ions, so we have the F1 particles, which creates a channel or a tunnel for the transfer of the protons, and thus it can easily go into the membrane, and where oxidative phosphorylation takes place.
Okay. Fine. So your test is over. Good. All the questions were answered well. Now Barbie will answer. Good. So Barbie now ready?
Yes, ma'am.
Okay. Now the first question from your cell chapter. Tell me that what is the structure of the chloroplast and how it helps in its function, which it performs.
Ma'am, chloroplast has outer membrane, then inner membrane. Inner membrane has the matrix or the stroma, which is the homogeneous fluid that contains starch grains, thylakoids, and many more. And thylakoids are the sac-like structure. Um, then come the granular thylakoids, which are arranged in a pile of coins. And then sorry.
Okay. So what function? Can you tell me the important part or important structure or structural component of the chloroplast? What is the function carried by the chloroplast?
Uh, it helps in the photosynthesis.
It helps in photosynthesis. Okay, so which is that part which starts the process of photosynthesis? In the chloroplast, which is the structural component which will initiate the process of photosynthesis? And where? Okay, which pigment is present in the chloroplast?
Chlorophyll.
Chlorophyll. Okay, where is this chlorophyll present? In the chloroplast. The location of the chlorophyll pigment in the chloroplast. I want to know
In the,
that.
in the thylakoid.
In the thylakoids. Can you further specify where in the thylakoid? Okay, so leave it. I'll take up the next question. From the biomolecules part, what is the difference between the aldose sugar and the ketose sugar?
Then allose sugar, mo- uh, monosaccharides are divided into can be divided into the carboxylic group, that is the yellow sugar and the ketose sugar. Yellow sugar has the aldehyde and ketose sugar has the chitin.
Huh? This classification is done on what basis? Aldoses and ketoses.
Carboxylic group.
Carboxylic group. Okay, now my next question from the cell cycle. Okay, what is chiasmata and when and how is it formed? What is
Kind,
huh?
the two homogeneous sister chromosomes, they cross over on the position called the chiasmata. It is in the meiosis for phase 1 in the archaea.
In prophase 1, in which substage?
In the third it is the archaea.
Again.
Then archaea.
No. Okay. So now can you name the substages of the prophase 1? Substages of prophase 1 of meiosis.
One is the liptolamin, then is second is the zygotenum, then third is the hacienda, then it is the diplotenum, and fifth is the
Dikinesis. Okay. One more question I want you to answer from this chapter. That is, what is the difference in the cytokinesis process seen in plant cell and animal cell?
Ma'am, can you repeat the question?
What is the difference between the cytokinesis process which occurs in the plant cell and the animal cell? Cytokinesis in plant cell and animal cell. How do they differ? Okay, now from respiration, that is your cellular respiration. Okay, now tell me, where does the glycolysis occur and what is the significance of it? The glycolysis process.
Ma'am, glycolysis occurs in the cytoplasm. Glycolysis is a process in which 6 carbon atoms converted into 3 carbon, that is the pyruvic acid.
Okay, correct.
And it is and then pyruvic is converted, oxidation, carboxylation process is there, so the pyruvic acid needed from the glycolysis process. So it is very important for the cellular respiration.
Cellular respiration.
And glycolysis is the first step in the cellular respiration.
Okay, any other significance of it? Can you tell me the net gain of the ATPs during this process?
Um, 2 ATP net gain.
2 ATP will be produced. There is a net gain of 2 ATP. Okay. Can you tell me the first step in the glycolysis or the glycolytic pathway? The first step in the glycolytic pathway.
Um, Glucose is converted into 2-phosphor. Um, um,
The first step in the glycolysis. First reaction occurring in the glycolysis. Okay, now tell me that total how many NADH2 are formed in the Krebs cycle. When one glucose molecule will enter into the Krebs cycle, or the product of one glucose.
NADH2.
NADH2, how many total NADH2 will be produced in the Krebs cycle?
Now in the Krebs cycle, 6 NADH2 will form.
Acha. Okay, so these are formed by the entering of one acetyl coenzyme A or two acetyl coenzyme A. Can you clarify that?
Now 1 acetyl coenzyme.
Okay, ठीक है. So your test is done. Now I would like the next person to get ready. Okay, Shravani now. Shravani,
Yes, ma'am.
you ready? Now, first question for you from the cell chapter. Tell me what is the difference between the eukaryotic cell and the prokaryotic cell?
Ma'am, the eukaryotic cell is the multicellular cell, and the prokaryotic cell is a unicellular cell.
No, itself is one cell. Okay, you can't say that this is unicellular or this is a multicellular cell. Cell is one cell. Okay, just think and say. Okay, move to the next differences.
Ma'am, this prokaryotic cell is found in the 70th type of ribosome, and the eukaryotic cell is found in the 80th type of ribosome.
So cell is found in the ribosome.
Sorry, 80th type of ribosome is found in eukaryotic cell, and 70th type of ribosome is found in prokaryotic cell.
Okay.
Ma'am, in a prokaryotic cell, the chromatin threads are not there, and in eukaryotic cell, the chromatin threads are there. Then in
So chromatin thread is only not there in the prokaryotic cell. So what is the genetic material then in the prokaryotic cell? Just clarify this difference, what you said. What you mean to say by that, that chromatin is there, chromatin is not there. You said chromatin is there in prokaryotic, it is not there, in eukaryotic it is there. What do you mean to say by that? Can you clarify it? So what is the genetic material in both the cases?
Ma'am, 1 second, can I think for
Ha ha.
1 minute?
Okay. Okay, I can give you next
Ma'am,
question.
can I tell one thing? Ma'am, prokaryotic cells are found in bacteria, and the eukaryotic cells are found in human beings or the organism like animals or this. Then, ma'am, in prokaryotic cell, there the nucleus is not present, and in eukaryotic cell, the nucleus is present, like nuclei and the membrane bound organelles are there, and in prokaryotic cell, the membrane bound organelles are they do not have any membrane bound organelles.
Okay, so instead of nucleus, what is present in the prokaryotic cell then?
Ma'am,
If
nu-
nucleus is not there, so then what is present? Because
ma'am, is it
nucleus, huh?
ma'am, is it nuclei?
Nuclei. No. Okay, now next question, Shravani, for you from your biomolecules chapter. Tell me, what is a triglyceride?
Ma'am, triglyceride is the combination of three fatty acids plus one glycerol. So the three fatty acids combined with the carboxylic group, that is three carboxylic group. So three the formula for the, ma'am, can I write and show it from here?
Ah, why not? Why not? You can show. You can tell me the formula of glycerol. What is the formula of glycerol?
Yes, ma'am.
Huh.
Ma'am, 1 second. I'm just completing that.
Don't write the whole equation. It will be
Okay, ma'am.
better then. I just want you to explain that the which functional group of the fatty acid is combining with the glycerol, and in the glycerol where is it combining? So if you will be telling me the formula of the glycerol, it will be clear.
Okay,
What is the formula, molecular formula of glycerol?
ma'am, it is CH3.
Okay, just leave it now. Tell me which bond is formed in the formation of the fat molecule.
Ma'am, ester bond.
Okay, so it is, how is it formed?
Ma'am, ester bond When three fatty acids, when combined with one glycerol, the three carboxylic group of the fatty acids, they combine with
Mm-hmm.
the the hydrogen or the sorry, hydrogen, uh, the atoms of the
Hydro-
atoms of the
hydroxyl group of the
hydroxyl group of the glycerol molecule. So
glycerol.
the bond form is ester bond.
Okay, ठीक है. Can you tell me the significance of the facts? Significance of facts.
Among facts, when they provide us,
That is the biological significance of facts. Huh. Okay, just one last question for biomolecules only. I'm telling, asking you that what is the difference between the saturated and unsaturated fatty acids? Okay, leave it. Now, last question. Total, how many amino acids are there in nature?
ma'am, in nature 20 amino acids are there, on
How
which
are they classified? How are they
ma'am,
classified?
10 are essential fatty acid, sorry, amino acids, and 10 are non essential amino acids.
Huh.
Like
So,
essential amino acids are used in living organ
huh? Huh.
living
Can you tell me?
organisms.
So, which one you call essential and which one you call non-essential?
Ma'am, the essential fatty acids, sorry, ma'am, the essential amino acids are that are as amino acids which are used in our body or in a human body.
Okay, leave it now. So, we come to the next chapter, that is the cell cycle. Uh, Tell me, what are the stages of the mitosis?
Ma'am, mitosis is divided into two stages, that is interphase and
The stages of mitosis, I'm asking.
sorry, ma'am. Ma'am, mitosis is divided into three phases, that is G1 phase, S phase, and G2 phase.
Think and say properly. Simply the stages of mitosis you have to name. Okay, I'll come to the respiration chapter now. So, in that, tell me, what is oxidative decarboxylation and when does it occur? Oxidative decarboxylation and when does it occur? Can you tell me the steps involved in the cellular respiration?
Yes, ma'am.
Uh, Tell quickly.
Ma'am, the first step is glycolysis.
Uh,
Ma'am, then Krebs cycle. or the tricarboxylic acid one, or
Tricarboxylic acid cycle. Huh?
acid cycle, then the uh, etc.
Okay. So, what is the product of glycolysis?
Two pyruvic acids.
Okay. Then, in the Krebs cycle, the pyruvic acid will enter directly.
Yes, ma'am, but it will first get converted into the coenzyme acetyl coenzyme A.
Okay. So, what is that process of formation of acetyl coenzyme A called? Okay, leave it. Now tell me when the NADH2 enter into the ETC, how many ATP molecules are produced?
3 ATP molecule.
When one NADH2 enters into the ETC, how many ATP molecules are produced? Okay, so your test is done.
2 ATP.
Now the next Rumisha will get ready. Okay, your test is done. Yes, Rumisha ready now?
Yes, ma'am.
Okay. Now the question for you from the cell chapter. So last student for the test and everyone try to listen carefully. So from your cell chapter, the first question for you comes, that is, what are oxysomes? Where are they present?
Oxysomes are the F1 particles. They are present in mitochondria in the inner membrane, which is having a finger-like projection, and finger-like projections called cristae. And there are small pigments called F1 particles. They are only known as oxysomes.
Oxysomes. Okay. What is the importance of these oxysomes?
Ma'am, they help in ATP. They help in production of energy.
Production of energy. Okay. So what process is going on there?
ATP synthesis.
ATP synthesis occurs during which process?
No, ma'am. It is having. It stores the energy. F1 particle stores the energy, which is in the form of ATP, and provides energy when it is needed.
Okay, so but how that energy, how that ATP is coming? From where that ATP is coming?
Ma'am, after the cellular respiration, when glucose is getting converted into ATP.
So when this glucose gets converted into ATP?
When it passes through glycolysis, Krebs cycle, and etc.
ETC. Okay. So can you specify out of this what you have named the steps in which case the ATP will be produced? The maximum number of ATP produced during which step?
Ma'am, please repeat.
During which step you have named just now the three steps? Okay, of the cellular respiration. In which step you find the maximum ATP are synthesized?
Ma'am, in Krebs cycle.
In Krebs cycle, the bag. So Krebs cycle directly the ATP synthesis takes place.
No, ma'am.
How many ATP are synthesized in the Krebs cycle? Can you tell me?
Thank you, ma'am. Ma'am, just a second.
Yeah, yeah.
3. Ma'am, 24.
24. Direct ATP you are getting in the Krebs cycle?
No, ma'am, we are not getting directly ATP in Krebs cycle.
How you are getting them?
Ma'am, by the conversion of NADP, NADH2 and FADH2 and GTP and ATP,
Huh?
then it passes through ETC, then the whole ATP, I mean that whole ATP molecule is formed then after that.
Okay. When FADH2 enters into the ETC, how many ATP molecules are synthesized?
Ma'am, 2 ATP molecules are synthesized.
Okay. ठीक है, तो I have asked the respiration part also here. Is it clear? That question is also coming.
Yes, ma'am.
So now one more question from respiration only I am covering up here itself. That is where the Krebs cycle occurs.
Ma'am, it occurs in mitochondrial matrix.
Mitochondria. Why does it occur there only?
Sorry, ma'am, this I don't know.
Okay. And what is the first compound formed in the Krebs cycle and how is it formed?
Ma'am, first compound is the citric acid, which is formed when acetyl coenzyme A combines with oxaloacetic acid, which is a 4-carbon compound, and acetyl coenzyme A is a 2-carbon compound, and citric acid formed is a 6-carbon compound.
Okay, good. So your respiration part is covered up. All questions well answered. Now, cell chapter, one more question I would like you to answer. That is, what are vacuoles?
Ma'am, vacuoles are the fruit packet. Vacuoles.
Just simply tell me the structure.
Ma'am, vacuoles are the circular one or oval shaped structures.
Okay. Bounded by how many unit membranes? Bounded by how many unit membranes?
Here's the definition.
Ma'am, I don't know.
Okay, leave it. Done. Now, tell me what is the difference between aldoses and ketosis?
Yes, ma'am.
Aldoses and ketosis, what is the difference between the two?
Okay, ma'am. Aldoses is a sugar which is present in aldehydes, and ketose is a sugar which is in the ketones.
Clarify that. What you mean to say present in the aldehydes and present in the ketones? What do you mean to say by that?
Ma'am, that means that aldehydes are producing aldose sugar, and ketones are producing ketose sugar.
Okay, further, what else difference?
Ma'am, aldehydes functional group is CH2, and in ketones it is OH.
OH. Ketone group is OH group. Okay, so now I'll take up to the cell cycle. Okay, now tell me, what are the stages of the mitosis?
Mitosis,
Yes,
prophase, metaphase, anaphase, and telophase.
telophase. Okay, before entering into the mitotic division phase, how the cell prepares itself?
Ma'am, just a second. Yes, ma'am.
How the cell prepares itself?
Ma'am,
Before it enters into the mitotic division phase.
ma'am, first enters into the interphase
Huh?
where the cell prepare itself for cell division by where the cell is metabolically active,
and
हाँ.
cell growth also takes place, and DNA replication also takes place.
Okay, but I want the explanation part कि कैसे क्या हो रहा है, किस समय हो रहा है, some further in detail.
Ma'am, first of all, when the cell, ma'am,
How
before
the
it,
interface? What is interface? Explain it
ma'am,
further.
it is a resting phase. It is a
हाँ.
resting phase which is present between the two successive M phases, where
Okay.
the two successive M phases where cell prepare itself for further division, and
Okay.
the cell is metabolically active, and DNA replication also takes place, and cell growth also takes place, but cell do not divide. It just prepare itself for division.
How many stages it is divided into the interface?
Ma'am, three. Main are three, and if we count the stage, then there are four.
Name them.
Ma'am, G1 phase, gap 1 phase, then S phase, synthesis phase, and G2 phase is there. And if we count the question shape, then G0 phase is also there.
Okay. So which phase is the longest one? Out of these, what you have named just now, which phase is the longest? And what happens in that phase? Okay, leave it. Just tell me whatever phases you have said just now, specifically what changes occur in each phase.
Ma'am, in G1 phase, the cell.
Okay, so your test is done now. Okay, so your test is done. I have already asked the respiration part. So all the students now, this way you must have seen the other children, the 4 students are remaining. Next biology period, you will be preparing yourself for the oral test. 4 children have completed, that is Neha, Barbie, Shravani and Rumisha. So your test is done. So start preparing for your MCQ now. Very soon I'll tell you that date. Okay, so before
Yes, ma'am.
Diwali only, I'll take up your MCQ also. So prepare it well. So we'll do Shanti part now. Oh,
Ma'am,
Shanti, Shanti, Shanti. Sai Ram, children. Yes, want to ask something?
Ma'am,
Yes, yes.
Ma'am,
Yes, yes.
ma'am, what about our marks?
ma'am,
Marks I will be telling all together.